Now, in the future, my view is we're going to need to be able to analyze proteins from 340 million American patients, or I don't know what Canada is now, 30 million or something like that, 40 million. And you're not going to do that with mass spectrometry. So together with Jim Heath, we've worked on microfluidic chips that have the ability to make miniaturized assays, ELISA assays using pairs of antibodies that can measure quantitatively proteins from very complex mixtures. So we now have a chip which in fact can make 50 measurements in 5 minutes, and it can analyze proteins down to a very, very low level. And we've shown that this chip, in fact, is working beautifully in the clinic. So what I'd like to be able to do is move from 50 measurements to 2,500 measurements, because I'd like to do 50 organ-specific proteins from each of your 50 major organs, and I'd like to do that in a longitudinal analysis for all patients. So you will serve as a control for when you're well, and you'll serve as a control for seeing when any organ deviates into a disease state and so forth. Now, the other thing that we've done is actually created a series of clinical assays at ISB, and I'm only going to tell you the general category of these different assays. So we've created a series of genomic assays, we've created a series of new proteomic assays, and we've created single-cell assays, and finally we're using iPS cells in really interesting ways. Anyone who's really interested in any of these, I'd be glad to go into them in detail, but remember these assays because their use is going to come up later when we talk about how to bring P4 medicine to patients. But what I will talk about is how we can use iPS cells to stratify really complex genetic diseases. And to give you an idea of what we mean by stratification, if you look 60 years ago, blood cancers were one disease, and 50 years ago they were 2 diseases, and today there are actually more than 100 different diseases. And the important point is we need to know which of those diseases you have, because generally each disease requires a unique therapeutic approach. So we're interested then in using iPS cells. We're collaborating with a company called Cellular Dynamics that can take 5 mils of blood from an individual patient and create induced pluripotential cells. And then they have the capacity to differentiate those pluripotential cells to neurons at 99% purity. So that means we can take neurons from any individual and put them in a test tube. And what we plan to do then in terms of Alzheimer's is 1, to get families, 2, to create the iPS cells, 3, to differentiate them in the test tube to neurons, and then 4, we'll hit those neurons with various environmental signals, natural ligands, drugs, RNAi. The idea being that for each subtype of the disease, you've disease-perturbed different combinations of networks, and hence the signals those cells deliver will be uniquely different for each different subtype of disease. So that's a hypothesis. We haven't yet proven it, but I think it's very likely to be true. This means that we'll be able, for Alzheimer's, to create subtypes, and then we'll be able to go back to family genomics and begin looking for the genes that encode those discrete subtypes. So I would say that all of these things we've talked about, family genomic sequencing, where we integrate genetics and genomics, this human proteome project, where we have assays for human proteins, the clinical assays, and finally a big new project we're working on with the world's 3rd largest database, that is to be able to capture all of the complete human genome sequence and their attendant phenotypic data, medical and molecular, and mine them for the predictive medicine of the future. And there are lots of barriers to doing that, quite obviously. But all of these things then take us to what we call P4 medicine. And let me give you just a quick, brief scenario on the 4 Ps. So for predictive, I've really told you major things that I think are simple and straightforward. In the future, you'll have your genome sequences done. We'll know an enormous amount about actionable genes that you have, and we'll be able to give you deep insights into how you should shape your behavior to optimize your wellness. And we'll do that on the individual basis. Number 2, I think we'll have a little handheld device that will prick your thumb and make these 2,500 measurements of the organ-specific proteins, and you'll follow across your entire life the evolution of any descent from wellness into disease and be able to react to it at a very early stage. The personalized is really all about the fact that we differ genetically on average by 6 million letters of the DNA language or so. So we're unique, we're different. I could never understand in medical school how you could argue based on population statistics from humans if we were that different. And it turns out I was really right. That is, as we'll see in a few moments, one of the big flaws of current evidence-based medicine and why I think it's come to the end of what it can do. But what it means is not only will we have billions of data points for each individual patient, we'll have hundreds of millions of patients with billions of data points that, again, we can search for the predictive medicine of the future. What's preventive about? Well, preventive is about a completely new approach to identifying drug targets. And that is, can you identify drugs that can go to the most proximal of the disease-perturbed network in a given disease and cause it to behave in a relatively normal fashion, abrogating the downstream deleterious effects? That's really a hard thing. Re-engineering networks is a hard thing to do. We'll have to learn how to do it in microbes, and we're making real progress there, and then later transfer it on up. Taking a systems approach to the immune system, for the first time we'll be able to understand effectively how to induce cell-based immunity. That's the big failure in all of the AIDS vaccines so far. And in fact, I remember arguing with Tony Fauci, who's the head of NIAID, the agency that's responsible for spending an enormous amount of money in AIDS, 10 years ago, saying, don't pour money on vaccines where mostly pharma companies have done it the same way Jenner did in 1796. Rather, spend just a little bit of money on a systems approach to understanding the immune system. And they're starting to do that now. And I think in a relatively short period of time, we'll have solved that problem. But from the preventive point of view, by far the most important thing that's going to happen is we're going to begin to focus increasingly on wellness. And I'll say more about that in just a few moments. What about the 4th P, the participatory? That is the real challenge. How do we bring and educate patients or physicians or the medical community as to the nature of P4 medicine? Physicians are really conservative. And getting them to change, I think, is going to be an enormous challenge. And we'll tell you at least one approach we're going to take to that in just a few moments. But on the other side of it, how do you create an IT for healthcare system that can deal with the network of networks that you have to be able to analyze for your patients? Show you a picture of that in just a moment. But let me say a word about evidence-based medicine. It's been productive in the past, but I think it has come to a point where if we pour increasing resources into it, there'll be an enormous law of diminishing returns because it's reactive rather than proactive. It's all about the disease rather than the patient. It's all about doing clinical trials on incredibly large populations of unstratified patients, as opposed to what P4 medicine proposes to do, stratify the patients such that you can go to the FDA with a clinical trial of 50 patients, and if 96% of them are successful, that will give you the statistical power to have the FDA approve them, and they did that for a cancer drug called Herceptin. Genentech did that. So I would argue that what really differentiates P4 medicine is it employs this network of networks. We have a genetic network at the DNA level, a molecular network, a cellular network, an organismic network, and finally the network of individuals interacting with one another, and all of these, of course, interacting with the environment. What is really terrific about networks is they organize information, they integrate information, and ultimately they are predictive in nature. So the power of networks, and as we know, networks have transformed how we think about finances, how we think about communications, how we think about many things, and now it's going to transform how we think about it. The two essences of P4 medicine are our ability to quantify wellness and our ability to demystify disease. And let me say just a word about wellness. Among the assays that we're developing now, we're thinking about assays that will let us, number one, quantify where you are with regard to your envelope of wellness, and number two, determine whether the slope of your wellness is toward greater wellness or away from wellness or dawdling in the middle. So this ability to assay wellness is, I think, as we'll see in just a moment, going to lead to a real transformation in interesting possibilities and so forth. So let's talk then a little about these four societal implications. One, I'd argue that the entire healthcare industry is going to have to rewrite their business plan in a major way over the next 10 years. And I would argue that most of them are going to really have a difficult time doing that. I'll tell you, in the 5 paradigm changes I told you about earlier, in every single case, we had to create a new organization to realize the paradigm change. It couldn't have been realized in the context of an old organization. Point number 2 is we're going to have an incredible digitalization of medicine. And that is going to occur on many levels. For example, how do you handle the billions of data points? Clearly, that's a prime example of digitalization. But even more, we're going to have sensors that will allow us real time to visualize enormous numbers of our own digital parameters so we can real time have feedback on our behavior and use that to optimize our behavior for wellness. I had a friend, Larry Smart, UCSD, who spent the last 10 years gathering longitudinal information on 65 parameters on himself. And what he learned from those studies was really revelatory, but the most interesting thing he learned is in several cases that doctors poo-pooed things where he was absolutely right. So one of the things that I would argue is really going to happen in the future is P4 medicine is going to be driven by patients in the future. It's going to be driven by patients. It's going to be driven by social networks. And it's not going to be driven the same way it is today by doctors and to a certain extent the health system. So how are we going to go about this P4 medicine? What are the real challenges for P4 medicine?